today's X-Talks webinar, Implementing Adaptive Trials, Operational Considerations. My name is Michelle Tran, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and includes a Q&A session with our speakers. It is designed to be interactive, so please feel free to submit questions and comments for our speakers throughout the presentation. Use the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message through this chat panel. At this time, all participants are in a listen-only mode, and the presentation slides will advance automatically. Please note that this event will be recorded and made available for future download. At this point, I'd like to thank IDDI, who helped develop the content for this presentation. The International Drug Development Institute is an expert center in biostatistical and integrated e-clinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI has offices in Belgium, Boston, Houston, and Cupertino in the United States. Now I'd like to introduce our featured speakers for today's event, starting with Mark Boyce, Doctor of Science, founder of IDDI. Mark holds a doctorate degree in biostatistics from the Harvard School of Public Health. He is the founder of the International Drug Development Institute and of PluPoints, and an associate professor of biostatistics at Hasselt University in Belgium. He has published extensively on cancer clinical trials. Our second speaker is Linda Danielson, Master of Science and Chief Operating Officer at IDDI in Belgium. She obtained her Master of Science degree in 1990 from the University of Wisconsin-Madison. She has over 20 years of experience in the pharmaceutical industry and worked at UCB prior to IDDI. She has also been president of the biostatistical section of the Belgian Statistical Society and the Belgian representative to the European Federation of Statisticians in the pharmaceutical industry. And now, without further ado, I'd like to hand the mic over to Mark. You may begin when ready.
Hello, everybody. Mark Boyce. Nice to have you on this webinar. And the webinar, as you know, will be on implementation of adaptive designs, operational challenges. I will start the webinar with a general introduction, and then Linda Danielson will discuss a case study, and I will close the seminar with some conclusions. So perhaps the best way to start is to define what we're going to talk about. We're going to talk about adaptive designs, and these are defined in the FDA draft guidance on adaptive design clinical trials as a study that includes a prospectively planned opportunity for modification of one or more of the study design features based on analysis of usually interim data from subjects in the study. In some rare cases, the adaptation can occur at the time that the final analysis of the trial would have taken place, but this has to be prospectively defined. So adaptive designs indeed use the accumulating data from the trial itself to adapt design features, and those include, and we will get back to this, the choice of a dose, the sample size, the interim analysis plan, the patient population, the primary endpoint, etc. As a matter of fact, recently, statistical theory has evolved in such a way that the major requirement of adaptive designs, which is that the alpha level or the type 1 error level be preserved. This is the main concern that the regulatory agencies have, that whatever adaptation you do not inflate the type 1 error, and that needs to be preserved at 5% usually, or less than 5%. Now, this problem is solved statistically. We now have methods that allow the alpha level to be preserved, whatever the adaptations, even very complex adaptations can take place, and yet the alpha level is preserved. And so we believe that attention should focus more on the logistics and interpretation of the results of adaptive trials rather than on the alpha level preservation, which is now well known. So we won't discuss methods to preserve the alpha level today. What we will discuss instead is really the logistical and practical aspects of implementing adaptive designs. Before we go into the case study, I wanted to discuss the kinds of design features that can be adapted in an adaptive design. So I've listed here the main design features of a trial, and obviously these design features include the treatment, which is under investigation, and the control group, the randomization ratio between these two options, sometimes more options than two, the treatment effect that is anticipated, and of course therefore the sample size of the trial that is based on that treatment effect of interest, the population of patients who need to be entered in the trial, the type 1 and type 2 error rates, the primary endpoint, the timing of the analysis, the primary statistical test, and the schedule of patient evaluations. Now, of all these design features, the most commonly adapted are the treatment itself in dose-finding trials. And we will discuss a case study of a dose-finding trial, a phase 1 trial in oncology. The randomization ratio can be changed adaptively, and that is done through adaptive randomization. The sample size is commonly increased in adaptive designs, and so we will very briefly touch on sample size increases. And finally, the population which is entered in the trial can be changed in an adaptive fashion, and that is done in enrichment design trials. If we look at the phases of development that are implicated by these changes, the dose-finding trial would actually be relevant in phase 1 and phase 2 situations. The adaptive randomization trials usually concern phase 2 trials, and then the sample size increase and the enrichment design typically would bear on phase 3 designs. Now, as I said, we will discuss today a very specific case study because we felt that it would be easier to discuss implementation issues if we were talking about an actual trial rather than in general. And the case study that we will actually look at is a phase 1 clinical trial. Before we move to the case study, perhaps a few generalities of why adaptive designs can be useful. Adaptive designs can be useful to handle uncertainty. It's fair to say that when you start a clinical development, particularly in phase 1 and even phase 2, there is so much uncertainty about the effects of the drug that it may be useful to adapt with some data at hand. And so we think that adaptive designs have a particularly interesting place in phase 1 designs, and that's why we chose this example of a phase 1 trial that uses an adaptive design rather than a fixed design. There is a need for flexibility in clinical trials. For example, it may be efficient to start several doses or several schedules of a treatment and to drop those that seem to be less efficacious, and this would be typically done in phase 2. Limited financial resources is another reason why adaptive designs are very actively studied, and this is particularly true of biotech companies who may not have the resources to do a large-scale phase 3 trial, but who may have the resources to do a modestly sized phase 2 that might actually lead into a phase 3. In that case, it is sometimes interesting to start a randomized phase 2 trial that is continued as a phase 3, rather than to do a phase 2 trial which is then stopped and then the phase 3 trial has to be started afterwards. And finally, and perhaps most importantly, we live in a fast-moving environment. New drugs get developed, new treatments get implemented or approved, and therefore for any trial of long duration, it's very useful to have the possibility of adapting the design of a trial given the changing environment. And that is sometimes done in phase 3 trials. So these are the main reasons why adaptive designs attract so much interest. And in spite of all these interests, the use of adaptive designs is actually quite modest. And why is that? We will get back to this actually towards the end of the presentation, but for now, I think the main reasons why adaptive designs haven't been used as much as they could have, and the reasons for that lack of interest, in a sense, are listed here. Actual gains from adaptive designs, in fairness, are generally small, so you shouldn't expect that the adaptive design will completely revolutionize the way in which drugs get developed. They are very useful to optimize drug development, but they won't change fundamentally the drug development paradigm that we've all been operating in. The theory of adaptive designs was until recently not so widely understood. I think it's fair to say that today there are enough fundamental publications on adaptive designs that the basic problems of them have all been addressed statistically. Sometimes people feel that an adaptive design is not as credible as a fixed sample design, and that may be true unless the adaptive design is completely controlled, completely pre-specified, and well executed. Some people have concerns that there is less regulatory acceptability of adaptive designs, but this I don't think is true. Both the FDA and the EMA have emphasized the need for adaptive designs and the fact that they would look at them very favorably if indicated. But then finally, the fifth reason for why adaptive designs are not implemented as often as they could or should is in fact the operational challenges that one is faced with when implementing an adaptive design, and this is exactly the topic of our talk today. As I said, we will use a very specific example of an adaptive design to sort of talk about all the problems or most of the problems that may be met when implementing an adaptive design. And we're going to focus on a very specific case, which is a phase 1 design using the CRM or continual reassessment method. The CRM is essentially an adaptive design that can replace, in many cases advantageously, the classical 3+3 design that in fact is used in 99% of current phase 1 trials. So currently most phase 1 trials in oncology use the 3+3 design to determine the maximum tolerated dose. And yet there is a vast amount of literature, statistical literature, that shows that adaptive designs, for example the CRM or some other adaptive designs, have better statistical properties and desirable operating characteristics as compared with the 3+3 design. So the question, to us at least, statisticians, is why do people not use an optimal design when they can, instead of using the classical 3+3, which does have major problems? And the two reasons why the 3+3 remains the norm is, of course, the tradition. You know, people are comfortable with 3+3. They know the design, even though it's not the optimal design. And then again, the operational complexity. Doing a CRM implementation, we will indeed cover these operational complexities and discuss them in detail. Before I leave the floor to Linda, I just wanted to introduce the concept of a CRM design. In a CRM or continual reassessment method design, one actually postulates a dose toxicity model, which is shown graphically here. And what you see here is 4 different curves corresponding to the same model but with different parameter values. The parameter is denoted beta, and so depending on the value of beta, you see that the dose response curve or the dose toxicity curve is moving to the right or to the left. So if beta takes a value of 30, a large value, then we see that the dose toxicity model shows much less probability of toxicity for higher doses of the drug than if beta is a small value, like 1. The 1 value is the top curve here, the blue curve. And so the first thing one has to do when implementing a continual reassessment method model is to ask experts from the field what is their assessment of the most likely dose toxicity curve. In this particular case, in the example that we will discuss, the dose toxicity curve that was estimated or anticipated by experts was the green curve. The green curve was such that for the second dose of the drug, 33% of the patients would experience a dose-limiting toxicity. And that, by definition, is called the MTD, the maximum tolerated dose. So in this example, the maximum tolerated dose is dose number 2, and at that dose level, 33% of all patients will experience, on average, a dose-limiting toxicity. And so the recommended dose for phase 2 is going to be the dose level just under that dose level. So the CRM works in this way. A patient is assigned to the dose whose probability of toxicity is closest to the target. So the first patient would be assigned dose number 2, because that's the dose at which experts anticipate to see 33% of toxicities. The patient is then treated at that dose level and observed to experience a DLT, a dose-limiting toxicity, or not. Based on this observation, you can then update the posterior distribution of beta and recalculate the expected value of the dose toxicity model. And then the next patient is treated and the process is repeated until some criterion is met. So one loops through this for all the patients, and the dose is increased or decreased depending on the CRM algorithm until we meet a certain condition. For example, that there is 90% probability that the next 4 patients will receive the same dose according to the model that one uses. And if that criterion is met, then the study can stop. So this design is of interest because it's completely pre-specified and one knows exactly when it will stop. And the advantages of this design, as shown on the next slide, is that in fact you don't waste too much time increasing the dose initially. By the way, this is an actual egg study, but it's not the example that we will discuss today. It's an example of another drug that was developed using the CRM. And as you see, in fact, the dose was increased quite fast. In fact, the first 3 dose levels were only tested in one single patient. So the first patient received dose level 1, the second patient received dose level 2, the third patient received dose level 3, and then the fourth patient received dose level 4, and he or she had a dose-limiting toxicity. At that time, the algorithm decided that the dose should be reduced, and so the next patient received again dose number 3, and so on for the next patients. So the little red dots here represent dose-limiting toxicities, and the blue curve represents actually the pattern of the doses given for the different patients in the trial. And so not only was this design interesting because fewer patients were treated at low doses, but also more patients were treated at doses close to the MTD. So with the CRM design, in a sense, there is no need for an expansion cohort, which would be based on some arbitrary number of patients, because here the expansion cohort is part of the design itself. And that's another advantage of using a CRM. So this is a sort of theoretical introduction to the CRM, and now I'm giving the floor to Linda, who will describe the actual case study that we would like to discuss.
Thank you, Mark. So as mentioned, we will be talking about a specific trial, but we won't go into the details of the trial. But the trial that we're using as an example is a phase 1 trial testing an add-on drug in patients with cancer. The patients were randomized double-blind in a 1:3 ratio to placebo or a dose of the experimental drug. And one thing I want to mention is actually the fact that the patients were randomized is not so common in a phase 1 oncology study. So that was one of the first unusual things about the study. Also, it was double-blind, so the control group, the patient and the investigator didn't know what they were receiving. It was randomized in a 1:3 ratio because, of course, we wanted most of the patients to receive the active dose. The DLTs were adjudicated by the Data Safety Monitoring Board, and because we were looking after each patient, we had to stop recruitment during each DSMB review. So the patient was recruited, they were followed up, we looked to see if there was a DLT, and then DSMB looked at it. I have a flowchart next slide to show a bit more. The DSMB could confirm the doses, and in our particular study, in fact, the design had to be revised during the trial. As Mark mentioned in his slide, we have to estimate what do we expect this curve to be, how do we expect it to behave to be the maximum tolerated dose. And in fact, the drug was much safer than they had estimated. So the protocol was revised 3 times to increase the dose to which we could go in the model. So we have a new patient enters the study. They're randomized to either placebo or a dose. Now, this is a bit different than your classical randomization because the dose is determined later in the model. So you don't know, you know at the time of randomization what the dose is, but you don't necessarily know what the next dose is. Then the site is provided with a kit number because it's double-blind. The patient outcome is entered into the EDC. In this case, it was toxicities, so safety data. The DSMB had to adjudicate also these toxicities because they had to determine, was this truly a dose-limiting toxicity? Then they gave feedback to the site. This information went into the clinical database, and then the CRM will take the data that we have so far, propose a new dose. Then the DSMB is involved again because the dose is not automatically changed according to the model because the DSMB has a final say looking at the safety and the efficacy. Do they want to use that new dose or do they want to do something different? And that's very important in this type of model, that you've got some control besides the automatic calculations. So in order to have something like this, of course you have to plan. In every trial you have to plan ahead, but specifically in an adaptive study, you need to put into place a cross-functional adaptive trial team. Ideally, you should have statisticians, trial managers, people in charge of randomization, people in charge of drug supply. You've really got to cover all the functions, and it's great if some of those people have experience in adaptive trials. We have to define the functionalities that are required in our system. Then, in order to be efficient, we need to develop custom software and links between the CRM, the IWRS, and the EDC system. There should be some communication system in place. As I mentioned, the sites had to stop enrolling while the DSMB was evaluating. So they had to know, yes, I can randomize, no, I can't randomize. They didn't need to know about the dose, but they did need to know, yes or no, I can randomize the next patient. We have to set up different committees and partners. An independent statistical center, because we're looking at the data on an ongoing basis, a DSMB, drug supply center, and others. And we need to facilitate the operations at the site and the DSMB level, because they might not be so used to adaptive designs. So up front, we need to help them so that it's easy to work. Part of it was the randomization. If it's adaptive, its centralized randomization is obviously essential. You've got to have all of the information in a central location. You should choose the randomization method based on sound statistical experience. Of course, you need to ensure the integrity of treatment allocations. But we all know that sometimes things happen not what you expect. They might give the wrong dose. They might use something that they shouldn't have. So that needs to be defined up front. How are we going to handle these deviations? Ideally, you should have in place statistical monitoring of the treatment allocations. And a 24/7 help desk is always very important for centralized randomization. But for adaptive studies, it's essential because things have to go quickly and decisions have to be made very quickly. In our case study, we had statistical involvement in all aspects of the randomization. There were statistical checks of the randomization at the time of the study setup. There was statistical monitoring of the balance and the allocation throughout the study. And all modifications were verified by the statistician. As I mentioned, we had to adapt the protocol several times. We had to update the model. We had to make some new assumptions. And that was in the CRM model, but it also affected the randomization. So everything had to be validated during the study. For the CRM, again, we want to choose experienced providers, and they should be involved early. They need to be involved, of course, in the design of the protocol. In order to know what we want to do, we have to perform different simulations to document the operational characteristics of the design. Information from the DSMB must be fed back into the system so that the CRM has that information and can calculate the next dose. Of course, key events and transitions between the different stages have to be well documented, and extensive testing must be done with active involvement of all key actors. Before you start the study, we have to have a lot of dummy runs. For the IWRS, obviously you need to choose experienced vendors and involve them early. The IWRS system needs to be integrated with all of the other systems so that things can go smoothly. Also, as we mentioned, the opening and closing of the randomization before and during the DSMB meetings. There had to be automatic communication with the sites. In our case, the randomization system would send an email to the sites letting them know randomization is open or randomization is closed. Obviously, we have to have good communication with the DSMB. And you've already seen this bullet, but we have to perform extensive testing with active involvement of all key actors in dummy runs. For the EDC system, the first bullet you've also seen again. We need to choose experienced vendors and involve them early. The EDC needs to capture real-time information on treatment and outcome. In this case, it was the DLT. And in fact, the DLT is not just one variable because it's based on adverse events. So the sites had to be encouraged to enter the data on an immediate and ongoing basis. Also, the data was used by the CRM for dose selection and review by the DSMB for the dose approval. And finally, also for the EDC system, we need to provide extensive testing with active involvement of all key players. And in fact, the EDC system, the IWRS, the CRM, the statistical modeling, everything has to work together. For drug supply as well, because drug supply for an adaptive study is not straightforward. You know, for a regular two-arm randomized study, you might have several doses, but you know exactly what the doses are up front. So if you can find vendors who have experience in adaptive designs, you should choose those vendors and, of course, involve them early. The packing and the shipments need to adapt as well, because you don't necessarily know up front what you're not going to need at the sites. Ideally, the packaging should also ensure that the blind is easily kept even after adaptation. In our case, the study drug was actually in syringes. So the syringes were prepared by an unblinded pharmacist who drew the appropriate amount of study drug and added the appropriate amount of sterile water. That way, when the investigator got the syringe, they just saw the syringe and they didn't know how much was active, how much was the sterile water. This was very good in our case because since the doses were adapted even more than we expected, having this possibility to add more or less liquid gave us a lot of flexibility. Of course, at the very beginning of the study, you need to optimize drug supply across all possible scenarios. And as we adapted our adaptive design, we had to also validate and optimize the new drug supply scenarios with the new maximum dose. And of course, we have to perform extensive testing with active involvement of all key actors. Our Data Safety and Monitoring Board, the key issues to consider, and this is actually true whether it's an adaptive design or any other study, your DSMB members should really be based on experience and independence. Some of our sponsors want to take the key opinion leaders, but if they don't have experience in a Data Safety Monitoring Board, and ideally some experience in adaptive designs, it's going to be very difficult. In the case of our study, we also had to detail how the role of the DSMB went beyond just the safety review. They were looking at the safety data, but they were also in charge of saying, is the dose suggested by the CRM the dose we want to use, or do we want to change it? All of this needs to be explained in detail in the IDMC charter. Again, all situations should also be pre-specified because we want our DSMB to be comfortable with what they're doing. We want them to have as little uncertainty as possible and know what to do in different situations. The sponsor, of course, has a role in adaptive designs, but ideally they should completely trust the DSMB and other committees with responsibility for recommending adaptive changes in open trials or implementing them in blinded trials. In the ideal world, the sponsor should not be involved at all. However, the sponsor often wants to be involved and sometimes must be involved if we're looking at a different type of adaptive design where we might be increasing the sample size. That has budget implications, and so obviously the sponsor has to have some say in that decision. So we have to make a rationale for involving the sponsor or not. There should be some strong and documented rationale for a few sponsor representatives to be involved. In fact, a few, I would ideally say one at the most, either to reach the best decision for the trial itself or possibly to secure further funding. But those representatives must be independent from the trial conduct. They should not be in any way involved in trial operations. They must clearly understand the issues and risks associated with knowledge of interim results. They need to be sure not to share these results with anyone else at the sponsor, even by nonverbal signals. So they have to be very, very careful. Again, minimal data should be shared. So the sponsor representatives should receive minimal pre-specified interim data. So only at the adaptation point. You know, in our study, the DSMB didn't need to recommend to change the sample size. The sponsor had to be involved because they saw themselves that we didn't have as many DLTs as expected. So they didn't have to have a representative involved in the DSMB. But sometimes we have to have that. But the DSMB should see more data than the sponsor. Of course, the documentation is key. Any release of information to sponsor representatives must be duly documented and tracked using a secure electronic system. And finally, we have to be sure that we have the adequate firewalls to guarantee blinding for all individuals other than those involved in the adaptive decisions, at the sponsor, at any CRO, at the sites. As I mentioned, in our study, we did have an unblinded pharmacist. It was the only way that we could do that drug the way we needed to. However, that pharmacist was not involved in any way in the study, so that was not a problem. Quality assurance, we need to think about system validation. In adaptive designs, there are many more different scenarios that you have to test and to consider. The validation can be useful also to double-check your assumptions and plausible scenarios. In order to validate, you have to try to think of all possible scenarios, and in this validation process, you might see that some of these scenarios don't go how we expect. Simulations need to be part of the validation, and of course, we have to have full documentation, both of the systems and the trial conduct. System tests. We need to test every system component separately and generate the testing documentation. So every system component, if you have IWRS, that needs to be tested separately. The EDC system needs to be tested separately. However, you also need to test the overall system in conditions that are as close to reality as possible. In the simulations, for the CRM, in our case, you need to simulate many trials to generate empirical distribution of desired operating characteristics for the sample size, the mean and the range, the proportion of patients treated at each dose level, the proportion of patients treated at doses above the MTD. This all needs to be done with simulation. And this simulation report could be considered to be a regulatory document in addition to your statistical analysis plan. So in summary, the flexibility of adaptive designs offers definite advantages, especially in early trials, phase 1 and 2. As Mark already said, the actual use of adaptive designs is limited, in part because of implementation challenges. Adaptive designs require careful planning, simulations, testing, and a choice of experienced vendors and committees. And above all, people working on adaptive designs have to be ready to adapt as well. Now, we've talked a lot about the CRM, but Mark will also talk about some other popular adaptive designs, so I'll pass it over to him again.
Thank you, Linda. And so let me, after this very detailed case study, let me just talk about a few other adaptive designs that have become quite popular. And I'm going to discuss, not without going into any details, but I will just discuss briefly the 4 types of designs that seem to attract the most attention. Seamless phase 2 and 3 designs, which is really a transition between phase 2 and 3, rather than doing phase 2 and 3 separately. Sample size increases, enrichment designs, and adaptive randomization. So going to the seamless transition first, I would recommend reading this very nice paper that describes how this seamless transition can be done. If we first look at the classical situation, the one that was prevalent until recently before the adaptive design sort of changed the situation, in the classical setup, we would actually do a phase 2 trial first, and then the phase 3 afterwards. The phase 2 trial would have as its main purpose to choose the dose. So maybe we would start a phase 2 trial with 3 doses versus a control. The purpose of the phase 2 trial would be to decide which of these 3 doses is the best dose to push forward to phase 3, and let's assume it's dose C. And so the phase 3 trial would then start after the analysis of the phase 2 trial was completed with dose C, which was decided to be the best versus control. Now, this classical paradigm worked really well, but it had a problem, which is that you had to actually wait here until you had the full results of the phase 2 trial before the phase 3 trial could be launched. Now, as we all know, it becomes more and more difficult to implement clinical trials, to launch a new protocol, to get the protocol accepted by IRBs and all the other bodies. So I think there is a tendency for people to try and combine different phases of research in a single protocol these days to sort of shorten this review time and acceptance time. So one way that can be done is what is called an operationally seamless phase 2/3, where in fact instead of doing two different protocols, one for phase 2 and one for phase 3, you actually start a trial that is a combined phase 2/3, which actually contains a phase 2 stage with the 3 doses being tested versus placebo, so the exact same comparison as before, but based on an interim analysis, one of the 3 doses is selected and pushed forward in phase 3 versus the control. Now obviously one hopes that the decision made on the interim analysis is as good as, and it's not always the case, but is as good as the final decision that would have been made if you had the full data from the phase 2. But oftentimes this is possible. Even with an interim analysis, one can already decide which of the 3 doses is the best to push forward. If it's not one single dose, then you could actually push forward 2 doses and drop one later in the development. So in a sense, the adaptive design framework gives you a lot of flexibility as to how many doses you test until you know that one of them can be dropped from further development. So this design is called the operationally seamless phase 2/3 because in fact you decide to launch the phase 3 based on an interim analysis of the phase 2, and so you've gained a lot of time. You've gained the time that would have taken to finalize the phase 2 analysis. You've gained that time, but unfortunately the patients who were entered in the phase 2 design may not be included in the phase 3 analysis. This is a model that the FDA likes very much because the phase 3 remains completely independent from the phase 2. And so this is the model that is most used today. There is, however, a more aggressive model in which the phase 2 part of the trial is also part of the final analysis. So here you have to do the transition between phase 2 and phase 3 adaptively. And you have to include the patients from the phase 2 in the final analysis, accounting for the fact that you started the trial with 3 doses versus control, and you kept only one of the 3 doses in phase 3. So that needs to be done in an adaptive fashion, and the testing procedure in this case is a lot more complicated than if you do simply the operationally seamless transition. But this is an option that perhaps needs to be discussed with the FDA and of course approved by the FDA or with the EMA and approved by the EMA. The gain that you might get from such a design is not just the gain in time, but also the gain in sample size, because now you've included the phase 2 patients in the phase 3 analysis as well. And so now you gain twice, you gain in time and you gain in sample size. So that's why these designs attract a lot of attention today. And again, we have methods, statistical methods, to control the type 1 error in these seamless transition designs. Another type of design that is increasingly considered is the adaptive sample size increase. And like I said, very often this is something that small biotech companies consider because they may not have the budget required to do a large-scale phase 3 trial. So the budget they have is enough to do a phase 2 randomized study, and you can actually do such a study and have a provision for calculating an interim conditional power at some interim analysis of the trial, or even maybe the supposed final analysis of the phase 2 randomized trial, and then look at the conditional power that that trial might have if you were to continue it beyond the sample size that's already accrued. If the conditional power is greater than 0.8, 80%, or 0.9, or whatever power you want to achieve, then you can continue the trial unchanged. But if the conditional power is lower than what you anticipated, perhaps because the treatment benefit is less than what you specified, then you might consider a sample size increase. And what's very often done then is to pre-specify a maximum sample size increase beyond which you don't want to go. For example, if you start the trial with 300 patients, you might say that you are prepared to commit another 200 patients or 300 patients, but not more than that. And so if the increased sample size that you would need to keep the conditional power at least 80% is less than the maximum pre-specified, then you just increase the sample size and keep going. If the increased sample size is not less than the maximum that you allowed, then you need to consider stopping the trial for futility. It may be that this trial will never show the the kind of benefit that you were hoping to get, and therefore it's as good to stop the trial now rather than continue it. Now, this is a relatively simple situation where the decision is simply to continue, increase the sample size, or to stop. But sometimes there is a subset of patients who may benefit more from the treatment than all patients. In that case, it's sometimes advantageous to consider not just a sample size increase, but also an enrichment of the patient population. And this again needs to be pre-specified. You must have some pre-specified subset of patients in whom, and perhaps this is for a targeted treatment, this is a targeted subset of patients, a subset of patients who have a certain gene mutation or a gene overexpression or some feature that makes them potentially more responsive to the treatment of interest. And so then you consider to increase the sample size but enrich it at the same time and put more patients who belong to the subset of interest in the future patient samples. And that of course depends also on the conditional power calculation. So here, if you can't increase the sample size to a number less than the maximum, you consider enrichment. If you then can enrich the trial and get the conditional power that you need with a sample size increase less than the maximum pre-specified, then you just increase and enrich. Otherwise, again, you consider stopping the trial for futility. So these are two designs that are gaining favor in oncology. One, the first one, the sample size increase, because the budget oftentimes is insufficient to start a large phase 3 trial. And the second one, when there is a good reason to think that enrichment might increase the power of the trial. And also, of course, zero in on the label that the trial will that the treatment will get on the actual patient population who will in the future receive the drug under development. And finally, I wanted to say a word about adaptive randomization, and by that I mean outcome adaptive randomization. What we mean by that is that the randomization ratio, suppose you have a control group and an experimental group, so you need to randomize patients between the two. In a classical design, that's done with a fixed randomization ratio, 1-to-1, for example. That means, on average, the patients are as likely to receive control as they are to receive treatment. Now, sometimes the ratio is not equal to 1. It might be a 2-to-1 ratio. If you want to put twice as many patients in the experimental arm as compared with the control arm, you can randomize patients in a fixed ratio of 2-to-1. And so on average, then, the patients will be twice as likely to be randomized to treatment than to control. But there is a catch here in the adaptive randomization, whereby the ratio of randomization is actually dynamically changed over the course of the trial. For every new patient, we have to have the outcome of the patient, and depending on the outcome of the patient and the outcome of all the previous patients, the randomization ratio can be adaptively changed to favor the arm that seems to be doing the best. So this is sometimes called the play the winner approach, because you put more patients on the treatment that until now has been doing better. Now, this looks like a very attractive idea, and it has been implemented in some trials. Recently, trials like the BATTLE trial or the I-SPY 2 trial have used this approach, and there's been a big controversy in the statistical world as to whether this was desirable or not. Many clinicians like this feature because it makes the trial easier to explain to their patients, and many statisticians, in fact, oppose this design because it has a lot of drawbacks. So if you have any doubts as to whether to use an outcome adaptive randomization or not, I would refer you to this very nice paper published by Korn and Friedlin in the Journal of Clinical Oncology. And this paper very clearly concludes that adaptive randomization is not recommended. If you want to have more patients in the experimental arm than the control, then you can do this using a fixed 2-to-1 or 3-to-1 or n-to-1 ratio, but to change the ratio over the course of the trial in general is not a good idea. So let me conclude this webinar by an interesting survey that was published in this journal, which used to be the DIA journal. It just changed names, but the contents remained very focused on regulatory matters and statistical matters and clinical aspects of developing new drugs. So this paper actually contained 3 pieces of evidence: a literature search, a review of clinicaltrials.gov, which is the registry maintained by the NIH, and a survey by the DIA's Adaptive Design Scientific Working Group, a survey of users to see if they were more likely to use adaptive designs today than they were 5 years ago. So first, the literature search, and this contained 8 statistical journals and 5 medical journals. This was performed over an 11-year period, and what is very clear from that survey is that the number of papers published in the statistical literature with at least one adaptive design item in the paper increased quite substantially over this time period. What is a bit disconcerting is that in the clinical literature or the medical literature, there wasn't such an increase. So it's as if statisticians spent a lot of time developing methodology that in fact the clinicians don't want to use. Hopefully that will change in the future. If we look at the registry data though, the clinicaltrials.gov registry, again we see that there's been an increase in the uptake of adaptive designs from 2000 to 2007, but unfortunately there is kind of a decrease in the number of trials done with an adaptive design afterwards. Now the authors of the paper discuss several reasons why this decrease might be seen, in particular the fact that there might be a delay in reporting trials to clinicaltrials.gov, so these numbers have to be taken with some caution. But I think the bottom line of this slide is to show that adaptive designs attract a lot of attention, they are well studied, but their uptake is perhaps not sufficient today. Now the most frequent uses of adaptive design, as I just said, were sample size increases, treatment group changes, for example dropping doses in phase 2, randomization ratio changes, so these were the most commonly used adaptive features, and the most frequent barriers against the adoption of these designs were listed here. The fact that you have to have change management to adopt adaptive designs, the fact that you have to have upfront investment and planning, and I think Linda's presentation demonstrated very clearly that yes, there is an additional investment in time and resources when you want to adapt to implement an adaptive design. Education is key. People need to know what they're doing, and that requires probably a higher level of understanding of adaptive designs than if you use a very simple fixed sample design. The infrastructure, again, Linda described the kind of infrastructure that needs to be put in place for adaptive design, but in fact I think one could argue that any well-conducted trial will need that kind of infrastructure anyway. And then finally there is this myth about the regulatory acceptance of adaptive designs. I think it's a myth. If an adaptive design is well justified and well designed and well conducted, I don't see any reason why regulators wouldn't take it just as easily as a fixed sample design. And the proof of this, of course, is found in the regulatory documents, the FDA guidance, which is still in draft form today, and the EMA reflection paper, which tends to be a bit old too, but hopefully the agencies will come up with newer documents soon. Finally, if you are interested in more details about this presentation, we recently wrote a chapter for a book that will appear later this year. The book is edited by Whaley Hay, Jose Pinero, and Olga Kuznetsova, and it will appear hopefully in 2014, published by Springer Verlag in New York. So I would hope that this book will be available soon, and if you wish to see a draft of this chapter, please feel free to write to us, and we will be very happy to share the draft chapter with you. And with that, I think I will leave it to questions and answers if there are any.
Well, thank you very much, Mark and Linda, for that insightful presentation. At this point, I would like to invite our audience to continue sending in their questions or comments for our speakers right now using the questions window for the Q&A portion of this webinar. I have already received some questions, and so I will start with those. This first question was asked during Linda's part of the presentation. Along with DSMB, can we also have interim analysis based on enrollment for big studies, for perhaps every 50 to 100 patients? And let's see, we have your thoughts on that, Linda?
I'm not quite sure I understand the question, but clearly if you are doing any type of interim analysis, you need to have a DSMB in place. So the DSMB can study, can look at safety data, but of course the DSMB can also look at efficacy data. So an interim analysis are definitely efficacy data, and that has to be done by the DSMB and can't be done by anyone involved in the study.
Okay, thank you very much, Linda. The next question was asked during Mark's part of the presentation. What are the different kinds of risks that should be considered early on during an adaptive trial?
Oh, that's a great question, because we now live in times of risk-based monitoring and risk-based management and quality by design. So I think the advantage of doing an adaptive design, besides there are other advantages as discussed during the presentation, is that these adaptive designs in a sense force you to look at all the outcomes that are possible. Because the adaptation, of course, will imply different futures for the trial depending on the outcome of the trial itself. And so an adaptive design will require the sponsor and the company that implements the design to look at every possibility that there is. Will there be an adaptation? Under what conditions? What could go wrong in the adaptation? What are the risks, as you said, of the adaptation? For example, one of the major risks of adapting a trial and increasing the sample size, one of the major risks, which we didn't discuss today because that wasn't really the topic, one of the major risks to the agencies is that after the adaptation, the population of patients who get entered in the trial changes. The clinicians who partake to the trial know that there's been an adaptation, and that may induce a change in their behavior. They may begin to enter worse patients or better patients. It's hard to predict which way it's going to go. But that, of course, would constitute a definite risk. So one has to foresee that risk. One has to address it. One has to have a statistical plan in place to address the risk explicitly and be completely transparent with the agencies as to what was done exactly and whether that risk was considered. So that, indeed, I mean, I hope we haven't conveyed the impression that adaptive designs had absolutely no risk, but the risks will be studied and simulated and documented before the study starts. So that, I think, is a definite advantage of adaptive designs. It forces you to look at every possibility, and that may be less the case in a conventional design.
Great, thank you for that, Mark. For this next question, in the Phase 1 CMR example, where do the proposed curves pick anticipated MTD?
Could you, I'm sorry, I think that's a question for me. Could you repeat the question? I'm not sure I got it.
Sure. In the Phase 1 CMR example, where do the proposed curves pick anticipated MTD?
Where did the proposed curves
Come from? Where
yes. Sorry. Oh, how were these curves drawn and before the trial started? Yeah. Again, a good question. In fact, as I said, these curves are have to be based on expert assessment. So you have to, you have to ask experts what they think the dose toxicity relationship will be. And then based on the expert opinion, actually it's easy to fit a mathematical curve through their expectations. Usually the dose-response relationship looks very much like the smooth curve I've shown. And the only thing that's unknown, in fact, is not the shape of the curve as much as where the curve is located. Is the MTD going to be dose level 2, dose level 3, dose level 4? That's in fact something that the experts don't know. What they have a pretty good idea about is how steep the dose-response relationship is going to be. And so you have to ask them the question, and oftentimes when we talk to experts, they say, well, we have no idea, but that's the point of doing the trial, isn't it? You want to see what the DLT, or what the maximum tolerated dose is, and where DLTs will begin to kick in. So again, I think the CRM forces experts to sit down, think very hard about the preclinical experiments, draw this dose-response curve or dose toxicity curve, and then the mathematical model will allow you to shift the curve to the right or to the left. So that's an essential part of the design, and it can only be based on expert judgment.
Thank you for that, Mark. Our next question is, in an add-on trial, the dose of standard care can sometimes be changed. Is there an adaptive design to handle this?
Okay, I'll take that one. Again, the key thing in adaptive designs is you have to plan everything up front. So you have to allow for that possibility that the standard of care might change. The problem is that you don't usually know at the beginning of the study that it might change. So I have a hard time thinking of how we could put that into an adaptive design because it's something you know as the study's going on and not ahead of time.
No, I agree. I agree with you, Linda. This, in my opinion, would not be an adaptive change. Suppose that because of some toxicities, you have to reduce the dose level of the standard, the backbone chemotherapy regimen. That, in fact, doesn't affect the test that you are doing of your new drug. It affects the backbone chemotherapy. So what I think can be done in that case, based on the DSMB recommendation, would be to actually reduce, if it's indicated, reduce the backbone chemotherapy dose in all patients, both control and experimental group, obviously, and then do a stratified analysis at the end of the trial, looking at the cohort of patients who receive the higher dose of backbone chemotherapy separately from the cohort of patients who receive the lower dose of backbone chemotherapy. But in a sense, that has nothing adaptive in it. It's just a change in the protocol, but it's not adaptive in the sense that it doesn't affect the treatment comparison that is randomized.
Thank you, Mark and Linda. For this next question, in a dose escalation trial, PK data may be quite important. Are there models that incorporate PK?
Yes, there are. Again, we didn't cover these, but there are indeed, models that use pharmacokinetic data in order to find the maximum tolerated dose faster. And for example, one such design is the so-called accelerated titration design, where in fact you can use PK data to go faster if you feel that the first dose levels are relatively safe, and if PK data actually confirm that perhaps you can increase faster than is usually done. So there are definitely references on trial designs using PK data. Some drugs are more amenable to use of PK data than others. Cytotoxic drugs typically are just increased until you reach toxicities. But some other drugs, again, that may be more dependent on PK data, you can incorporate those data into the CRM. The CRM has led to a number of extensions. You can do CRM designs with 2 drugs, combinations, using PK data, using other levels of toxicity than the dose limiting, so lower levels of toxicity. There is a ton of papers in the statistical literature on variations around the CRM. It's a pity, really, that there are so many papers that describe how to do it and so few trials that actually do it.
Great, thank you for that. And I believe we have time for one more question. For a single-arm adaptive trial, for example, CRM, do you still recommend to have a DSMB?
I'll take that one. Yes, I would still recommend to have a DSMB because even if it's a single-arm study, we can have, you're trying to look at the dose and you want to have somebody independent to look at the dose. It might not be a classical DSMB because they're not looking at safety, but they're just looking at the CRM design, but they're still independent from running of the study. So I would say in all cases of adaptive designs, you either need a data safety monitoring board or possibly a special committee that's an adaptive design committee. For example, if you had a phase 3 study where you had a DSMB that was looking at the safety over time, but it was a phase 3 adaptive design, you might want to have another committee looking at the adaptive part of it. If the DSMB really felt more comfortable looking at safety, you might have a second independent committee to look at that adaptive part.
Thank you very much for that answer. And thank you very much to both of you for those answers for the Q&A. We have reached the end of the Q&A session of this webinar. If you have any further questions, or if you'd like the slides for this presentation, please direct them to Dominique Grisard. Her contact information is showing on screen right now. Now please join us in thanking our speakers, Mark Boyce, founder of IDDI, and Linda Danielson, Chief Operating Officer at IDDI in Belgium. Thank you to everyone for participating in today's conference. You will be receiving a follow-up email from XTalks with access to the recorded archive of today's event. A survey window will be popping up on your screen, and your participation is appreciated as it will help us to improve our future webinars. We hope you found this conference informative. Have a great day, everyone.